Hi, my name is Dean Mills, and my project studies the growth of Haloquadratum walsbyi, which is a halophilic archaea. Archaea are one of the three main domains of life, along with bacteria and eukaryotes. Archaea are single-celled organisms like bacteria, but have some components in common with eukaryotic cells. Here are the three domains labeled with halophiles circled. Haloquadratum walsbyi to the left is one of the most dominant halophiles in nature. It is one of the larger halophiles and can uniquely grow in a square shape. To the right is Halopharax vulcani, which is a smaller common halophile. Below that is Solenobacter ruber, which is actually a bacteria that can also grow in hypersaline environments. Some halophilic cells are photosynthetic and will turn a pinkish color. Examples of this in nature are in Nevada and Utah, where halophiles have been able to grow in abundance in high salinity environments. Many halophiles need environments far saltier than most ocean saltwater. Despite Haloquadratum walsbyi being one of the most dominant halophiles in nature, it grows slowly in a lab setting compared to Halopharax vulcani or Solenobacter ruber. The hypothesis for this experiment was that if components from H. walsbyi's environment were added to its media, then it would grow faster. So here's a visualization of the project. H. walsbyi was grown in its own media, and both H. vulcani and S. ruber were grown to mid-log phase before the cells were pelleted in a centrifuge and the remaining media was sterilized by filtration. 10 milliliters of that sterilized media was added to 40 milliliters of sterile H. walsbyi media. Then 1 milliliter of the H. walsbyi from its own separate media was added into the combined media. The H. walsbyi was grown in 250 milliliter flasks that were stored in a 40 degrees Celsius incubator. Growth was measured observing the optical density of the media with a spectrometer. At the bottom of this picture is a spectrometer along with a diagram of how it works. Also pictured is an incubator and autoclave, which works as a sterilizing pressure cooker for the media, and only some of the salt that went into 2 liters of H. walsbyi media. Here's a filter sterilizer in use. It's a filter designed to exclude anything longer than 0.2 microns in length. A centrifuge was used to pelletize the cells before the media was filtered. Also pictured is a comparison between two flasks, one with growth and one without any. You can see that the flask with growth has turned pink, like the halophiles pictured in nature. Here are some growth charts of H. walsbyi in different media. The x-axis in both is time, and the y-axis is optical density. Both the positive and experimental groups were grown in triplicate. The points on the graph are the averages of each measurement, and the error bars represent high and low measurement. Not graphed was a negative control that did not exceed 0.01 optical density at any point. The graph on the left shows H. walsbyi growing in either its own media, which are the blue points, or the media that it was in, or the media that was made in part with the H. vulcani treated media, which are the red points. You can see that the positive control did grow faster, but eventually they reached about the same optical density. The experiment for the S. ruber media was pretty similar. The graph on the right shows the H. walsbyi growing in either sterilized 40 to 10 media, which is the blue positive control, or the 40 to 10 milliliters of media where the 10 milliliters was treated, which is the red. Again, you're seeing pretty similar results. This tells me that adding the treated media of either H. vulcani or S. ruber did not significantly alter the growth of H. walsbyi, which goes against the original hypothesis. On this graph, we have X is time again, and Y is optical density. The data are the same as the last graphs, but using positive controls for each. The red points are the growth from the H. walsbyi in its own media, and the blue points are from H. walsbyi grown in media with sterile S. ruber media added. And despite this being in its own media, the media containing the fresh S. ruber has the H. walsbyi growing faster, which suggests that something in that media is causing it to grow faster. So to come back to my hypothesis, adding sterilized components of S. ruber or H. vulcani had little effect on the growth rate of H. walsbyi. However, supplementing the sterile S. ruber media into the H. walsbyi caused the H. walsbyi to grow faster. And if I had more time, it would have been interesting to retest the growth of H. walsbyi in S. ruber media more thoroughly and to stain cells and use a microscope to see if there are any other contaminants in the media, especially since H. walsbyi is fairly distinctive looking. I'd like to acknowledge the Mills Lab at Holy Cross for providing a space to perform this experiment and letting me use 30 flasks at the same time, Dr. Sparks for her input, and the NIH for providing Mills Lab with funding for my materials.